Welcome all, and thank you for joining today's AWRI webinar. My name is Robin Dixon and I'm a Senior Viticulturist at the AWRI. I'm joining you today from McLaren Vale on Kaurna land. In this session, we'll look at soil health, carbon and undervine management in a dry climate. In the spirit of reconciliation, the AWRI acknowledges the traditional custodians of country throughout Australia and their connections to land, sea and community. We pay our respect to their elders past, present and extend that respect to all Aboriginal and Torres Strait Islander people today. I would also like to acknowledge Wine Australia for providing funding and support for webinars via the AWRI Extension Project. Before we jump in and make a start, a couple of quick reminders to anyone who is new to the AWRI webinars. If you would like to provide a comment or ask a question, please click on the Q&A button at the bottom of the Zoom toolbar. So type in your question at any time and click send. We'll be holding the Q&A session at the end of the presentations. Also a reminder that this session is being recorded and a link will be sent through later to view via the AWRI's YouTube channel. We would also like to collect data today on your knowledge of this topic. You should see a 1-question survey pop up on your screen asking you to rate how much you know about soil health, carbon and undervine management in a dry climate. If you could please rate your knowledge on this topic on the scale between 0 and 10, with 0 being nothing and 10 being expert, and you should be delivering this webinar. At the conclusion of today's webinar and after the Q&A, we'll prompt you again with another single question to establish your knowledge on the topic after the webinar. Again, you'll be asked to rate your knowledge on the scale between 0 and 10. So for anyone who has just joined, welcome. Today's webinar topic is soil health, carbon and undervine management in a dry climate. It's a great pleasure to welcome Professor Tim Cavagnaro, Dr. Thomas Lines, Dr. Joseph Marks and Dr. Mardi Longbottom. First up, we have Professor Tim Cavagnaro. Until earlier this year, Tim was Professor of the School of Ecology and Deputy Head of the School of Agriculture, Food and Wine at the University of Adelaide. Tim's research focused on soil ecological processes with an emphasis on the impacts of land management and environmental change on soil ecosystem services. While Tim works in a range of agricultural systems, vineyard floor management, soil microbiomes and carbon sequestration are major themes in his research. Tim recently took up the position of Dean of Graduate Research at Flinders University and is also an adjunct professor at the University of Adelaide. So Tim, if you're ready to make a start, I'll hand over to you.
Great, thank you, Robin, for that introduction, and thank you for the opportunity to be here and speak today. It's fantastic to have this opportunity, and we always enjoy getting out and talking to people face to face, but we also really value these webinars and acknowledge the AWRI for providing these and Wine Australia for supporting them. So today we're talking about work that's covered in part through a Wine Australia funded project looking at cover crops and vineyard floor management and soil health. And so I'm going to give a little introduction, and then Tom will then move on and talk about some specific results from that Wine Australia funded project that we've recently completed. So as Robin said, I've recently moved institutions, but still very actively involved in this work. And my new contact details are up on the screen, and always happy to take a question via email after one of these events. So I guess if we're going to talk about soil health and soil carbon, we need to think a little bit about what is soil, and that might seem like a little bit of a silly question, but what I want to present to you is a soil ecologist's view of the soil environment. And so when I think about soil, I think of it as being the most complex material on the planet. And I will argue that point with anyone who wants to take me up on that. It is an incredibly complex material. If we simply look at these soil profiles from different locations around Australia, you can see a huge amount of variation and complexity. So in some soils, we have organic matter layers at the top. We might have bleached horizons. We might have leached horizons. We might have different texture contrasts, everything ranging from sands right through to very heavy clays. So there's a huge amount of variety among different soil types, but also as you move down a soil profile, there's a lot of diversity and change. Even if we look at soils on a relatively small scale, so here we might be looking at an individual farm scale, you can see that the person managing this particular farm has to contend with quite a few different soil types that you can see here with the different colors of those soils. And that's just looking at the surface. So there's going to be differences as you move down the profile as well. And that presents a great challenge to work with. And when I talk about soil being the most complex material on Earth, I mean that in the sense that it is a single material that contains the solid, the liquid, and the gas phases. So all in one, we have these very, very different components that make up this one single material. And if you were to look at a typical soil, and these are very average values, it might be made up of 40% of mineral materials. So that's the sand, silt, and clay particles. There might be about a quarter that's air and about another third that's water, and then a couple of percent of organic matter. The point is that when you look at soil, much of it's actually empty space. And that kind of blows my mind that when you stand on the soil, you're actually standing mostly on empty space, right? You have those pores and channels between the soil particles that contain water, or they contain air, or the gases. And the image on the right-hand side of the screen here is where a block of soil has been taken and then put through a CT scanner, much as you might encounter in a hospital for medical scanning. We can use those for soils as well. And the grey material there is showing the soil particles, but the rest of it is empty space. So again, what we have when we look at the soil is this complex material that contains the liquid, the gas, and the solid phases, and it contains a lot of empty spaces that form networks of tunnels that vary massively in their properties. So if you think of the soil, it's this really complicated material that contains all these interconnected tunnels and channels of different sizes and shapes. And so organisms living in that environment have to contend with that complexity. It also contains very diverse resources. So the soil is very much of a product of the materials that it was derived from. So the rocks and minerals that were weathered to form those different particles, and they all have different chemical and physical properties. And the other thing about the soil is it can change in a moment. Okay, if you think of an organism that's happily sitting in a little pore inside the soil, and then suddenly it rains or the irrigation is switched on, it suddenly goes from being in a gaseous environment to being completely flooded. And so that can change momentarily. And so the life in the soil has had to adapt to and contend with all of that change over very rapid time periods. And so when I look at the soil as an ecologist through the microbial ecologist view, with all of this diversity, you get more ecological niches. And where you have more niches in an environment, life has come up with different solutions to solving the problem of existing in that environment. And so that's why soil contains arguably the most diverse communities, biotic communities on the planet. Right? So if we were to take 1 gram of soil, get a teaspoon and take a gram of soil, it would contain billions of bacterial cells comprised of many, many thousands of different species. In fact, if you were to take a handful of soil, there are more bacteria or more organisms in that handful of soil than there have ever been people on the planet. I don't mean the population now, I mean the population ever. There are also many tens of meters of fungal hyphae, and those hyphae are really important, very fine filaments that grow through the soil, and they hold those soil particles together and help give it its structure. Also within the soil, we have many many centimeters of roots in that single gram of soil. I remember sampling an organic farm in California, and we found over 10 centimeters of root per gram of soil. Right? So we're cramming lots of life into all of those little spaces between the solid soil particles. Just to give you some numbers from a vineyard, this was some work from the Barossa at one of our field sites, and we found about a tonne of earthworms per hectare in the top 15 centimetres of soil. So to put that in other terms, that's the equivalent of about 20 sheep, all right, dry sheep equivalents per hectare. So when you look out across a field and you see the sheep, just remember the earthworms, all their biomass. And that was just in the top 15 centimetres of those soils. So again, huge amounts of life within the soil. It's a jungle down there. It blows my mind as an ecologist to look at all of the diversity that's down there and to try and explore and understand what that biology is doing. And so why does that matter? Well, the reason that this is important is because ecosystem services are the benefits that are provided by the natural environment. So the life in your soil is providing different services that we all rely on every day. They're cycling nutrients. They're taking nutrients in organic forms and degrading them or mineralizing them down into inorganic forms that can be taken up by plants and vice versa. The life in the soil plays an important role in purifying the air that we breathe and the water that we drink and we irrigate our crops with. This soil can contain many beneficial insects and invertebrates as well. The microbes play an important role in the delivery of nutrients. So I'm sure you've all heard of rhizobia and legume associations where they capture nitrogen from the atmosphere and they deliver it right to the plant in the roots. We have mycorrhizal fungi that can take up immobile nutrients like phosphorus and again, deliver them directly to the plant. Also, microbes and the life in the soil is central to carbon cycling. So when plants photosynthesize and they capture that carbon and it makes its way into the soil through organic matter or root exudates, it's the life in the soil that helps cycle and stabilize and sequester that carbon in the soil. And there's a whole host of other ecosystem services that the organisms in the soil provide to us. So what we're really interested in is thinking about how do we optimize our management systems to provide the microbes with the things that they want so they can then deliver us the ecosystem services or the things that we want. And it turns out it's pretty straightforward. They want the same things that we want. They want a source of food. They need a source of carbon and nutrients. They want some water as well. Water is fundamental to life everywhere, really, and it's especially true in the soil environment. They also want to be protected. They want some protection. They want to be managed in a way that minimizes the disturbance of them so they can provide those valuable ecosystem services to us. And that, of course, all relies upon the way in which we manage our soils. And so that's really the premise that lies at the heart of the work that we've been doing through this work, looking at vineyard floor management and undervine cover crops, is really, can we provide the food and the water and the protection to those organisms in the soil? And I guess the sort of overarching question that we're asking is, how do we bring the life back to the soil? And so that's really what we've been doing in this project over recent years, again, with very generous support from Wine Australia. And so at this point, I think I'll hand over to Tom, who will get into some of the specific results from one of our most recent trials. So thank you. Over to you, Tom.
Actually, I'm going to jump in and introduce Tom. Thank you, Tim. I feel like your presentation was a love letter to soil. I loved it. Thank you. And now I'd like to introduce Dr. Thomas Lines. So Tom is currently working at the University of Adelaide on quantifying the effects of undervine cover cropping. He finds motivation in providing an evidence base which growers can use to make informed decisions to improve their industry. Before Tom started in this field, he studied algal physiology, chemical engineering, and biochemistry. Tom, if you're ready to make a start, I'll hand over to you.
Thanks, Robin. Yes, that's right. And actually, I'll get a chance to speak a bit about algal physiology today, hopefully. So I'll just reshare that. Okay, that's working in full screen. Okay, yes. So thanks, Robin. Yeah, Tim's lead up there was really perfect, a perfect lead into this work, and the premise for trying to explore or trying to understand why cover cropping in the undervine space might actually be a very useful tool for growers of really any crop. So we've spoken on AWRI webinars previously about our undervine cover cropping work in Nuriupa and Langhorne Creek, and the logical next step was to take cover cropping and this method for trying to understand the effects to the highest producing region in the country, which is the Riverland. But that presents its own challenges. The environment's quite different there, the soils are different, so it really does justify a bit of work to try and understand what the best method is to try and incorporate a reduced herbicide approach to the undervine space, to try and provide the nutrients and the shelter and the water for microbes in the soil to be able to better provide that functionality that we know healthy soils can provide. So that leads us nicely into this project. So this trial has been going on for 2 years, and I'll jump into more of the details shortly. So we set up the trial at Monash. This is a map of all of our cover crop trial work and sampling work over the last few years, and a lot of the focus has been on this Monash trial site for the last 2 years. The trials that we tried to the treatments that we tried to incorporate were based on our previous work at Nuriupa and Langhorne Creek, and so we looked at medic and rye as an undervine, a really successful undervine treatment, and comparing that to herbicide as well as a few other methods. So I'll step through that now. So yes, the first, or the control treatment, is the herbicide, and this is what the vineyard looked like at day 0. So quite sandy clay soils. Actually, it did vary a bit between plots, and you can see there's not a lot of growth. Heavily sprayed and intensively managed over the years, and so the soil structure at the surface was really poor. When it would get wet, it would be really muddy, but there was very little growth, and provided a really interesting and different environment from the Barossa Valley. So for the control, we had a herbicide in the undervine, and then we tried to use the mid-row to generate biomass and side throw that biomass into the undervine space. So one of the concerns that we had going into this project was that we weren't going to be able to generate enough biomass in the undervine space alone. So perhaps we could just use the mid-row to generate biomass and side throw. So you'll see this is a bit of a trend in the treatments. The next treatments that I'm going to talk about are the medic and rye and the native grasses. So the medic and rye, we had 2 treatments there, one just undervine and then one mid-row and undervine. And then the native grasses, we put native grasses in both the mid-row and the undervine. And this is how we did it. I used a hand seeder, which I was much better using when I wasn't trying to fill myself. And then seeding natives, and Andrew Fanny came out to help us put in the native grasses, because those grass seeds are so light and fluffy, and very difficult to actually get in the soil with traditional seeding equipment. So we used Andrew's equipment there, and the horticultural sort of system, the market garden system to put in the medic and rye, and so those were our undervine treatments, and this is what they look like after a couple of years. This is just going into vintage last season. So, not vintage, going into the season. So you can see the medic and rye, really great mat of biomass there. It's really quite deep. That's just after 2 years, and it's the recurrent, or the succession has been really successful, and there's not a huge amount of new growth, but all of that dies off and mats down and provides this incredible protection for the soil underneath. So I couldn't believe how well that went. And I think perhaps this is one of the biggest, or the most important points, is that the medic and rye does tend to outcompete really everything. We had a few spots where fleabane was coming up in that, but not really much at all. And you could spot spray that if you wanted. We also put it in the mid-row, and in the first season we had a dripper line down the mid-row as well to try and make sure that the investment in putting seed in the mid-row actually paid off. So we had huge biomass down the mid-row, and then in the second season, we didn't irrigate because obviously that's not what, we expect growers to be doing. So we turned those drippers off and let rain do its thing. And we got biomass production, but not quite so much. And the purpose of having that mid-row, again, was to side throw it and place it in the undervine space to try and improve the mulching. But really, we didn't need to do that because, with the rainfall, but also with irrigation, the undervine cover crop actually established really well, but still died off really quite early. And that was for the medic and riot. When we're looking at the native grasses, they established really well also. The mid-row native grasses appreciated the water and they had a really good stand in the first season. The second season, where we didn't irrigate the mid-row, there wasn't anywhere near as much biomass produced. However, in the undervine space, the native grasses got going and have been successfully growing since then over the last couple of seasons. So definitely a feasible approach, and we'll get into some of the data as well shortly. We also trialed 2 mechanical weeding methods. So we had the knife weeder and the multi-clean. We wanted, because we're interested in the effects of soil health on the effects on soil health of these different management practices, different methods for disturbing the soil can have different effects on soil health and microbiomes and these sorts of things. So you can see the knife weeder, obviously cultivates at quite a low level. Is fairly effective at removing weeds, but does still require a few passes per year. Similar things with a multi-clean, but much less soil disturbance. So we're hoping to see a bit of a difference there between the two. And also the multi-clean can have a bit of a damaging effect to the trunks of your vines. It removes some of the bark, and with heavier, woodier weeds, like, again, fleabane is a common problem in the Riverland. It was a common problem everywhere. If you're just taking the tops of those plants off, they can come back bigger and better and cling on next season. So fleabane is probably an entire session on its own, really. But I want to get across that if you have the right cover crop, you're going to outcompete it. And fleabane in itself isn't really, the worst thing in the world. Obviously, you don't want a cover crop of fleabane, but it's one of those early colonizers that's just trying to take advantage of bare earth. And if you don't have bare earth, there's nowhere for it to live. So yes, we had mechanical weeding 1 and mechanical weeder 2, which is the knife weeder and the multi-clean. And so then we collected a heap of results on all of these things. We looked at yields and we looked at the soil. So we'll get into some of these now. So total soil organic carbon is one of those indicators of a whole suite of soil health sort of characteristics. So we talk about soil organic carbon, but carbon in itself isn't the most useful thing, but it provides a lot of good functionality. So if you look at 2022, you can see pointer options. I can do visible. You can see my mouse. So in 2022, we didn't see much of a difference, which makes sense because we'd only just put in these treatments. In 2023, we did see even more variation, but nothing significant. Perhaps an increase here of treatment at treatment E, which is the native grasses, and the native grasses continue growing all the way through the season. So those root exudates are going into the soil and building carbon consistently. We also looked at nitrogen in the soil. We did see a significant effect where treatment D had a higher ammonium content than treatment B and treatment A. So A and B, remember, are the herbicide controls, and C and D here are the medic undervine treatments. So we did see perhaps a bit of an increase in 2023 of ammonium. And then when we look at juice nitrogen, we didn't get a significant effect, but we did see perhaps the beginnings of an emergence of a trend, if you allow me to say that. So higher juice nitrogen without bringing that yeast assimilable nitrogen above 100 milligrams per liter is quite a beneficial thing. And if you can reduce your inputs in winemaking, you might be able to improve wine quality and things like that. We also looked at soil microbiome, and I thought we only looked at soil microbiome in the second year because we didn't want to waste resources on looking at this in the first year when we weren't expecting to see many results. So we looked at it in the second year, and saw very little difference between the treatments, and I was quite surprised. So we looked at bacterial populations and fungal populations. So that's the 16S and ITS. And we looked at a suite of different metrics, and we didn't really see much difference between them. These are the taxa bar plots. So when you assign on the tens of thousands of sequences, you try and identify exactly which species that they are associated with, and then you can look at who's who. And something that I recognized, and something perhaps that's really fortunate that I happen to have done a PhD in algal, like freshwater ecology and looking at algae, but I started recognizing these freshwater algal species, and specifically Microcystis, which is a cyanobacteria that actually produces an important toxin. And my hypothesis is that obviously, we had some severe flooding throughout the Riverland, and everyone was using the water from these floods. I think the microbial populations in the waters that were then pumped onto the vineyards and the orchards contained a huge diversity of microbes, which then sort of flooded the soil microbial populations. So when we looked at the DNA, we actually didn't see much of a difference. And Microcystis is specifically interesting because I had a look into the toxin microcystin that's produced, and if you apply that to vegetables and other plants, you can actually get translocation of that toxin from the soil into the fruit. It's not a big problem for grapes, it doesn't look like, but it's definitely something worth considering, and something I'm sure most people haven't thought about. We saw a significant effect on juice sugar in 2023, nothing in 22. Again, with the native grasses having a slightly higher sugar content than treatment F here, which was the knife weeder. This is the sort of effect that we've seen previously with the more aggressive grasses in the undervine space, reducing berry size potentially, though we didn't see that. And so, yeah, the beginnings of some trends that we want to look at into the future, ideally. Anthocyanins, we did see some significant effects here again, with the medic treatment D having significantly less anthocyanins than the native grasses or herbicide 2. But there's a lot of variation here. Did we see any effects on yield? No, we didn't see any effects on yield whatsoever. There's still a lot of noise in the data, so I just thought I'd show you a video of Joe picking some grapes. But yeah, early days. I think in the long term, and with some drier years coming up, we might start to see some difference. Yeah, another good reason to try and continue the trial. So I think with that, massive thanks to everyone who's been part of the project over the last 2 years, and all of the previous work in URI and Langhorne Creek. Joe as well, Tim for some great support and advice. Omar Najjar for offering his vineyard and providing so much time, and then everyone else who's been part of the project. We also generated a few YouTube videos, which these QR codes will link you to if you're interested in having a look. And then otherwise, happy to take some questions when we get to those. So thanks very much.
Great, thank you, Tom. We'll have to check out those YouTube videos. I guess we will be sharing them in our email that we send out to participants with the recording of this webinar.
That'd be great.
Yeah, thank you. Excellent. Now I'd like to introduce Dr. Joseph Marks. So Joe's background is in terrestrial ecology with a specific interest in soil-plant interactions. So last year, Joe completed his PhD?
Yes.
Yeah. So just, I think you told me the day before your twins were born. So congratulations on both of those achievements. Investigating the influence of undervine management on the ecological intensification of vineyards. Specifically, he focused on the influence of cover crops on arbuscular mycorrhizal fungi, soil organic carbon dynamics, and plant litter decomposition. His ultimate aim is to communicate with grape growers to better understand their challenges and convey the outcomes of his research to enhance sustainability within the Australian wine industry. So Joe, if you're ready, hand over to you.
Thanks, Robin.
Do this for you.
Thanks, Robin. Tom's just going to introduce the project that we've been working on very briefly, and then I'll get into some methodology and data. So, Tom.
Yeah, thanks, Joe. So, yeah, this presentation that Joe and Marty are about to give summarizes the results of a project that was sort of conceived a year or so ago now, where funded by the Friendship Fund, which is the AWRI and the University of Adelaide decided to put together some seed funding for projects to try and build the collaboration between the two organizations. So when we heard about that, we had a bit of a think about what sort of work we're doing, and what might be of interest to the AWRI, but also to the grape growing population as a whole. And with such massive interest in carbon sequestration and management practices, we thought we'd maybe be able to look at the intersection of these things. So yeah, we reached out to Marty and conceived this project, and this is work that Joe's done a lot of over the last year. And yeah, I think that actually I should also mention the Limestone Coast Grape and Wine Council. They'd heard about the project and wanted to extend it to their region. So they were able to use some of their regional funds to extend the project down to that region. So we were able to, vastly increase the amount of data that we collected through their contribution too. So thanks to them. I think that's enough for me. And yeah, thanks, Joe.
Thanks so much, Tom. And thank you, Robin, for that really nice introduction. Let's get into it. So always acknowledgements first. Big thank you, as Tom said, to the Friendship Fund for this collaboration, to the AWRI, Uni Adelaide, and Sustainable Wine Growing Australia, and the plethora of help we always receive, without which we would get nothing done. So I always start with this slide. It's sort of tried and tested, but I think it really hits home with regards to the importance of soil organic carbon. Tim touched really beautifully on it to begin with. But putting it in the context of this project, what I really like about organic carbon is the fact that it sort of ticks both these boxes of the economic and the environmental, often which sort of, I guess, butt heads, and unfortunately the environmental is often sort of put to the back when the economic comes to the fore. But with regards to organic carbon, we see both boxes ticked. Never more evident than with these sort of 6 points regarding soil structure, how it helps to improve porosity and aeration and root movement, water dynamics, the ability for water to become held in soil pores and be exchanged with plant roots, infiltration dynamics, cation exchange capacity, so the ability for soil to hold nutrients and then sort of exchange those with plant roots rather than them being leached. We see a positive feedback loop occurring with organic carbon, that being the more organic carbon you can build or retain, it helps to protect itself through the enhancement of soil structure and stability as well. And of course, we talk about biodiversity a lot these days, and it's so important. We're able to sort of quantify it much more readily these days and cheaply. And so biodiversity is certainly improved through organic matter retention and enhancement. Microbes, as Tim mentioned, love to consume them, and therefore those microbes then feed into the trophic connection of soil, leading right up to that sort of the macro faunal diversity. We don't see mackerel in there, but certainly worms and bugs that are fantastic for soil health. And of course, sequestering atmospheric carbon is really important. So we've looked at the benefits of organic carbon, but what about the benefits of planting cover crops under vine? And of course, they work together. This little schematic shows the difference, between one side with a cover crop under vine and the other side with a bare earth. And, some of the co-benefits that can come from having cover crops. Of course, cover crops do increase your organic carbon, and we're going to look at that later. And they also help to retain organic carbon by stabilizing the topsoil with an additional root system. But more than that, they also increase the infiltration rate of water into the root zone where it's needed, rather than staying on the surface, which you might see in more of a crusted herbicide managed soil. On the surface, it can be prone to evaporation, which is not beneficial to the plant. It could also cause runoff and lead to erosion. So getting into the soil is really important. The cover itself, the top cover, the shoots of the cover crop also act to disperse the energy of raindrops, which in a hot, dry Mediterranean type climate like we inhabit can be quite impactful, especially on sloping vineyards. And so a lot of research conducted in places like Sicily or Rioja in Spain, where hillside viticulture is super important. A lot of that research is concerned with topsoil retention through the planting of cover crops. So let's get into the project. The approach and methodology first. So we focused on two regions, the Barossa and the Limestone coast, both of which are pretty big regions, so huge spatial variation. And so in terms of choosing sites to look at, we wanted to choose sites with contrasting management practices. So a site A might have an undervine cover crop, a site B might have an undervine herbicide. Now, within these regions, there are vast amounts of variation in the soil, things that we simply don't know are there and could be masking our results. And so to sort of account for that and mitigate as best as we can, we would employ a practice where we chose paired sites as best as we could. So if site A was located here, we would try to find site B with a contrasting practice located within a similar spatial region, similar spatial context, which would then allow us to be a little bit more confident perhaps about, the fact that we're sampling from contrasting management practices and that the differences we see are from those management practices rather than from underlying spatial heterogeneity. We sampled about 20 sites per region, roughly equal numbers of contrasting practices, taking 3 cores per site. So it was really sort of a survey of these vineyards and these practices. So a bit of a snapshot first. We're going to start really broad and then go much more narrow and detailed. So from a broad perspective, we've got a couple of slides first. These are principal component analyses. The idea here is that you just plug in all of your data points, which each dot represents a data point. And then you can also plug in the variables that you measured, so pH, clay content, organic carbon, and then see how those data points are being pushed and pulled. And what you might expect to see is some separation of the two groups. So our groups here are Barossa and Limestone Coast, and we do see some variation. We do see a lot of variation. We also see some separation. That separation doesn't appear to be due to the organic carbon stock. Mostly it appears to be due to clay content, pH, things like that. And we would expect to see that because pH is higher down at the Limestone Coast owing to that limestone substrate. Another bit of a shotgun approach is to just put in the data points and group them by undervine practice. So cover crop, herbicide, or surface. Surface is either something, it could be a surface amendment, so compost or mulch, and then see how they fall out. Again, this isn't a super useful, picture. This shows a lot of variation. It shows a smattering of things. It tells us that we need to dig deeper and have a bit of a closer look at what's going on. So let's do that. So one thing we wanted to do was see what might be driving soil organic carbon, as well as, and also alternatively to the undervine management practice. And this is a correlation plot. So a correlation plot basically shows how much one thing correlates with another. The darkness, the more dark blue and larger the circle is, the more two variables correlate with each other. So obviously stock and stock correlate 100%. But if we have a look at soil organic carbon stock and go along the line, we see some increased correlations, but especially with nitrogen. So we know that nitrogen and soil organic carbon correlate very well together, as evidenced in the next slide. We also know that clay content can correlate with organic carbon. However, when we correlate clay content with total organic carbon here, we see a very weak correlation. Now, we know that to have sort of a 90%, 100% sandy soil isn't going to retain any organic carbon or hardly anything. But by the same token, having a 90% clay soil is just far too strong, far too dense to have any micropores to be able to hold your carbon. So again, the take-home message here is that there is a sweet spot with regards to soil texture and its ability to retain and increase carbon. That is to say, soil has a ceiling with the amount of carbon that it can retain and build. Back to my previous point regarding nitrogen, very strong correlation here between total nitrogen and total organic carbon. So 90% correlation, very strong. We know that soil microbes, in order to break down, to decompose, mineralize carbon, they need nitrogen. Without nitrogen, they simply can't function. So that correlation is really, really important. So let's get into some results. Again, starting very broadly. On your left, we see this is the limestone and Barossa data pulled together, and a box plot showing the herbicide versus the cover crops. The yellow line indicates the mean value, so that's probably the best thing to look at. And the different letters here show that there is a statistically significant difference. That is to say, there is more organic carbon under your cover crops than under your herbicide. Now that's true for both of the sites together, and especially true for the Barossa. Pulling the limestone coast out separately here, we see a difference, you know, and there is perhaps more organic carbon under those treatments, but there's something else masking it. Perhaps there's not enough samples. Perhaps we need to delve a little bit deeper into the limestone coast to see what's happening. These are all the sites together. And so what this shows is the huge contrast in organic carbon stocks in the different sites. So right down here, we see as little as 12 tons per hectare, right up to over here where there's at least above 80 tons per hectare. And I can tell you those two sites are from contrasting management practices. I'll let you guess which. But there is a lot of variation in between that data. Now, I think the most important thing to do now is we've taken this big, broad snapshot, and let's hone in on a real site-specific detail or a case study from each region. First one in the Barossa Valley. These are anonymous vineyards, so unnamed, but it is in the Barossa, this one. And this was a really, really great site because it was an individual vineyard site that had a vineyard with a long-term cover crop, a legume and a grass. And right next to it, 5 meters away, was one that has just undergone a heavy till and is undergoing a successional change. And that gave us a really great ability to try to test two contrasting practices within a very close spatial region. No guesses. The 12 tons per hectare belongs to the tilled site. The 36 tons per hectare belongs to the cover crop site. And so, perhaps that cover crop site has reached its ceiling. Yeah, they can't maybe get any more carbon into the soil. And so what this shows us is that, between these two practices, if you till your soil, if you disturb your soil, as Tim mentioned, you'll lose that carbon really quickly. 3 times more carbon in that cover crop vineyard. Let's have a look from the limestone coast. So another site-specific case study. So 3 vineyards spatially close. So this is a short-term trial that Tom has been conducting down in Padthaway. And what we can see here is some real contrasting carbon stocks from highly contrasting management practices. Again, these were all herbicide originally, and then they've been built up for over about 3 years now. We've got a herbicide, and that is 18 tons per hectare. And then we've got very subtle differences between these two practices, which are a surface mulch and then a surface compost amendment as well. So even in that small amount of time through that surface mulch, that perhaps increased infiltration and water retention, we've seen a much greater carbon accumulation, which is really cool to see. Some nice little take-home messages. Carbon in soil changes with many variables, okay? Some of these are in your control and some of them are not. We know that nitrogen is vital to see increases in carbon. And from previous research that we've conducted from controlled long-term trials in the Barossa and in Langhorne Creek, we found that the employment of a combination of a grass and a legume, in our case, a medic and a ryegrass, were really beneficial. The ryegrass is vigorous. It provides a lot of biomass into the soil, especially root content. But in order to do that, it needs nitrogen. And so planting it with a leguminous partner that provides a nitrogen source for that ryegrass is really beneficial, so that the ryegrass doesn't rob the grapevine of nitrogen. So that partnership works very nicely. And so, yeah, that leads on to the last point, which is the cover crop selection is vital to see a productive vineyard while also increasing carbon and doing what else other things you might want to do. So you might plant a cover crop to do one thing, but you're going to see a lot of other co-benefits happening as well. And I think it's really important to manage those expectations, to understand your terroir and to understand your targeted approach. I'm always aware of time, so I'll leave it there. And I think Marty's going to have a chat next. So thanks so much for tuning in.
Great. Thank you, Joe. I've been furiously writing some questions for you. So just a reminder for people who are watching, please send in your questions. You can put them into the Q&A section now, and we'll ask them at the end of all of the presentations. Thanks, Joe. Now I'd like to introduce Dr. Mardi Longbottom. Mardi began her career in the wine industry, helping to establish her family's vineyards in Panthaway in the early 1990s. She holds undergraduate and postgraduate qualifications in viticulture from the University of Adelaide and has extensive technical and vineyard management experience in Australia and the USA. For the past 14 years, Mardi has focused on wine industry sustainability projects, including regional climate risk analysis, benchmarking greenhouse gas emissions from vineyards, and the management of corporate water assets. She manages the technical and membership aspects of the Australian wine industry sustainability program, Sustainable Wine Growing Australia. So just before I hand over to Mardi, thanks for sharing the image. I think that's probably Tom Lines, is it? Doing the paragliding up there. We'll talk about that in the Q&A. But if you're ready, Mardi, I'll hand over to you.
Thanks, Robin, and thanks everybody for joining. I want to acknowledge at the outset this project that we've been doing with the University of Adelaide. Tom mentioned it before, but it's essentially funded through the University of Adelaide and the AWRI Friendship Fund. So what I'm going to present to you today is really the second part of the project that Joe's just presented you the results of the soil surveying that we did. But the second part of the project, which is where AWRI came in, was really trying to take the data that was collected in these soil surveys and try and work out how we could best use that data if we were to integrate it into Sustainable Wine Growing Australia. So just as a quick introduction, for those of you who are not familiar with the Sustainable Wine Growing Australia program, it is the national sustainability program for the wine industry. So members of the program go through a process each year of reporting through their data. So there's quantitative data across a range of their practices, and they also go through a self-assessment workbook, which represents qualitative data that we also collect. So the areas that we cover include things like soil and water management, and also greenhouse gas emissions. And then those who take on a bigger commitment to the program go through an independent audit process and become certified members of the program. Now, how this relates to this soils project is that what we wanted to do was really, based on the data that we collected, so through those surveys in the Barossa and Limestone coast, have another look at the data that we're collecting through Sustainable Wine Growing Australia and really work out whether there was some more metrics that we could be collecting, which would inform our larger data set. But then also, how do we take that data and integrate it into better decision support tools? So I guess in terms of the importance of soils and soil management to the wine industry, I think we all appreciate how important soils are to both soil health and vine health. And both Tim, Tom and Joe have really explained to us the importance of that. But also soil and vine performance and soils management is really important. This final one here that I've got, though, soil carbon sequestration, this is one that probably has been more highlighted in the last few years. And in terms of the number of inquiries we get around what is the ability for us to sequester carbon in our vineyard soils, this one comes up a lot and has generated a huge amount of conversation. So this is the bit that I think probably we can extract and really make use of this when we're looking at Sustainable Wine Growing Australia and that data. So as an industry, we have a goal to get to a net zero emissions position by 2050. And as a reminder of how that really works and what the steps that we need to take to get to the net zero position, the first thing we need to understand as businesses and as landholders is what our total emissions look like from our property. Once we understand our emissions, we have to be able to demonstrate that we're reducing emissions, and then we have to offset those emissions with something by a change in practice. And only then can we say that we are truly at a net zero position. So in thinking about that and thinking about soil and soil management, we're really looking and focusing at this carbon offsetting step. So once we've understood our emissions, reduced our emissions, there's a potential there to use soil carbon sequestration as an offsetting mechanism. Now, I'm not going to go into any of the details about that, but that's really what we're thinking about here. Now, luckily for those of you who are already members of Sustainable Winegrowing Australia and who are very familiar with the reporting that goes along with that, you will know that you're already collecting your Scope 1 and Scope 2 emissions, and that you'll get a report that looks like this one here on your screen each year, showing you exactly what your emissions are from your site and what the breakdown looks like across electricity use, fertiliser use and fuel use. So this is a typical profile here. And these numbers on the y-axis are kilograms of carbon equivalents. That's for the whole site. So once you've understood your emissions, it's then looking at, so what is the opportunity then to offset those emissions using soil carbon sequestration? And this is just so that you can understand, I guess, how this works. If you can increase your soil organic carbon by 1 tonne, this is equivalent to sequestration of 3.67 tonnes of carbon equivalents. So just keep that in mind when you're thinking about things that you can use to offset your emissions. Now, the current metrics that we collect, the first part of this was to have a look at what are the current metrics, review those and see if there are any gaps or opportunities to expand those metrics around soils. So we're already collecting quite a bit of information here that's relevant to soils. The climatic environment or the region that we're operating in, that can inform us about what the potential is for soil carbon sequestration, the management practices, how big the vineyard area is, whether or not people are using irrigation, how much biodiversity area they have dedicated in the property, and then how people are managing their mid-rows and undervine. So that's what's in the current data selection. And what we've gone through and said, well, actually, if we were to increase the metrics that we collected from members, there are things like these ones here that we might include in future years. So things like cation exchange capacity, individual cations that people are measuring in their soils, what type of soils there are, total organic carbon, bulk density, and then going into a bit more detail about what people are actually doing in their undervine and also mid-rows, and then how people are doing their soil sampling. So if we were to introduce those and collect them, it's then, so once we've got this data set, how can we better use the data set for not just benchmarking to start with, but also how do we use that to inform decision-making about what people are doing in their soils and vineyards? So members currently get a report that looks like this one here. This is just a snapshot of it, but this is the temporal benchmarking. So for the data that people are reporting each year, you can go back in time and see how you've changed with regards to your performance each year. So things like your yield, water use, nitrogen use, etc. And what the aim would be is that if we were to introduce these new soil metrics, that you could also do this same kind of temporal tracking for the soil components. So things like soil organic carbon, you'd be able to track that over time. And another example could be soil pH. But again, we're not absolutely sure on these metrics. This is just an example of what could be done. The other kind of benchmarking report is the ones that look like this, and this is where you can compare your own performance compared to other members of the program. And this is one of the examples that we look at greenhouse gas emission metrics. So on the left here, you can see the on-site total greenhouse gas emissions, and then the components of that being fuel, fertiliser and electricity use. And the idea here is that you look at the numbers here, this is a percentage and a ranking, and this says at 49%, you're roughly in the middle compared to all of the other members of the program. Now, broadening that back to soil metrics, if we were to introduce a new vineyard soil measure, that measure could be potentially made up of things like, again, soil pH and soil organic carbon. So it's not just about looking at your own performance over time, but also comparing it to other members of the program. And then, so this is the data that Joe just showed you in the previous presentation. We've got this data that we've already collected in the surveys. It's about using that and obviously, I guess, understanding that there's a huge diversity in soils and soil components. But the more data you can collect, this can better inform what we do around soil management in vineyards. So it's expanding that with the data that might come through with people's reporting, and then us then developing a decision support tool within the program that's based on that data that we've got. So for example, if we were to introduce a drop-down menu here, you can choose, okay, well, what if I was going to do some compost application? That compost might be made up of different manures, mushroom compost, and different examples there. If we chose mushroom compost, and this would be based on the data that's coming through the program, go through that decision process, and ultimately what we'd hope is that that could give you an estimate of the total soil carbon sequestration that could be possible in your soils. So again, this is just examples of the things that we'd like to be doing. And as part of this project, it was to come up with some recommendations for where we might go next with the program. Coming back to this achieving net zero goal that the wine industry has, I think the other thing that's really important to acknowledge here is that this understanding our emissions profile. Now, as I said, members already have a good idea about this because they've been receiving these reports for a number of years. But one of the things that we did last year, and this is really a repeat of an exercise that we did back in 2016, was to take all of the data that we've been collecting through sustainable wine growing over a number of years and come up with what a typical emissions profile for vineyards and wineries in Australia looks like. Now, and I think there's a couple of really interesting things. One interesting thing is that there hasn't been a big change since 2016 to when we did this again last year. But if we look at the entire profile from Australian viticulture, the two big contributors to emissions in vineyards is from electricity and diesel use. So if you're looking for opportunities to reduce your emissions, these are the two areas that we have a lot of control over, and that they are things that we can reduce our emissions by changing our practices around electricity and diesel use. And just for interest's sake, this is a typical emissions profile for an Australian winery. And no surprises here, but the greatest source of emissions from wineries is electricity. So if you're looking at reducing emissions, these may be other areas where we have a bit more control and you can have a bigger impact potentially on your emissions from the vineyards and wineries. So that was the end of my presentation, but happy to take questions on that area.
Great, fantastic. Thank you, Mardi. So now I'd like to invite Tim, Tom and Joe to join us. We will start the Q&A session now. So just a reminder, if you have questions, just type them into the Q&A section down the bottom of your toolbar. We've got a few questions already. So for Tom, what was the sowing rate in kilograms per hectare for the medic and ryegrass mix, please?
I had a second to have a look just then. It was about 30 kilos per hectare. But you know, you could go a bit lighter or more, it wouldn't matter too much. And that was just done once and it self-sowed the next year.
Okay, excellent. Thank you. Mark Krasnell, our friend over in New Zealand. Hello, Mark. Hi, Tom. Cool stuff. Did you happen to find more of the algal species in the more herbicide-heavy treatments? So could the poorer soil structure have led to more waterlogging and hence algal presence?
Interesting one. I'm attributing the algal presence in this instance to the presence in the water that's been irrigated. So while we do see, you know, cyanobacterial slimes and things like that in the undervine space when there's bare earth and poor structure, and you can see that, you know, that mossy, slimy section underneath the dripper line often. In this case, I think it was coming straight out of the pipes and into the soil and essentially being filtered there by the soil. And you know, that's largely where we're sampling in the slightly more moist soil. So what would be interesting is to look at the degradation rate of that DNA over time, potentially, because you would think with a cover crop and more microbial activity, you should see a faster rate of decay of those things. But yeah, that's sort of my take on it.
Okay, great. Thank you. Now, a question for both Tom and Joe, I think. How is mulch how does this carbon sequestration of mulch compared to cover crops?
You want to take that?
Yeah, it's a much, I guess, in terms of the stratification of the soil, it's much more on the surface. And so, you know, it's really hard unless you're mechanically getting the mulch down into the soil. You're not really going to find a lot of carbon perhaps below depth. So a lot of that's going to be on that real surface area. Being that it has a sort of a higher C to N ratio, it's going to take longer to break down by microbes. And that in itself offers, I guess, there comes the co-benefits. So it's retaining more moisture. It's adding a protective layer. It's reducing temperature. So a lot of other things going on, which could potentially be increasing the microbial diversity below a little bit as well. And of course, it's prone to no disturbance. So there could be other things going on. But certainly there's no active root systems, there's no active photosynthesis. So there's definitely a marked difference. And from previous studies, we have seen over a 7-year trial, a good sort of 15% more organic carbon in sort of active root-based cover crops than surface amendments.
Okay, thank you, Joe. Now, Tim, you'd like to jump in?
Yeah, if I can add a comment to that, I think, yeah, absolutely agree with what Joe said there. Another thing with mulch is if they have a very high carbon to nitrogen ratio, so they're very rich in carbon and low in nitrogen, when you add that to the soil, it can actually cause the microbes to compete with the plants for nitrogen. And so you get what's called a nitrogen drawdown effect. So you actually render some of that nitrogen less available to the actively growing plants. So it's, yeah, not all mulches are created equal as well, but thoroughly agree with what Joe said about stratification of that carbon. If it's sitting on the surface, it gets decomposed, it goes off as gas quite quickly.
Great, thank you. And just on that point, so Joe, you showed a graph where there was a strong correlation between nitrogen and soil organic carbon, but then it was interesting to see in Tom's results that the native grasses had the highest level of organic carbon and not the legume, so the ryegrass medic mix. And Tom, I think you mentioned that it was because you thought it was because the natives were there for a longer period of time, so the whole season. Do you have any other comments on that?
Yeah. Well, I think my take on that is that there's still nitrogen in the soil and the grower is still applying fertilizer as they normally would. So the fact that there's higher soil organic carbon in the soil, it's likely that, you know, the available nitrogen in the soil is being locked up potentially to some extent to fix that. But, you know, there is still variation within that C to N ratio that we talked about. It was about 10-to-1 in across the Barossa and the Limestone coast, and I'd expect it to be pretty similar in the Riverland. So yeah, it's an interesting point. I guess the takeaway is that, you know, you can still increase nitrogen, but it doesn't have to be available. So you know, your aggressive grasses could be the best way to increase your carbon stocks, but that may have other consequences. And we're trying to better understand exactly how more aggressive grasses in the undervine space actually do lead to reduced yields. I think it's quite a complex picture.
Okay, great, thank you. Now a question for Joe. How often would soil sampling be required? So a question for Joe and Mardi, and I'll combine it with another question. So soil sampling is costly in an environment where the industry is not doing so well. In a vineyard with different soil types, how many samples would be required to get an accurate level or measure of soil organic carbon?
Yeah, I mean, sampling, it's one of the great questions in science and certainly in sampling, sample-based science. It's very difficult to know exactly how many samples you need to get an accurate sort of measure of what's the truth. The truth is out there. What is it? In terms of carbon, it's a slow process. So in terms of the frequency of going out and sampling, you want to make sure that you're sampling at the same time point for sure. So, you know, if it's the start of the growing season or the end of the growing season, you want to make sure you do that whenever you resample. So I would say with carbon, maybe every 3 to 5 years. It's a slow process. You can lose it really quickly, but it takes a long time to build. So, you know, yeah, and so it really is a balance between, yeah, how much money you have available, the resources, and it really does come down to that sort of that baseline logistics and a trade-off really between, you know, accuracy and precision. So really one needs to be intelligent about where they sample, stratifying if it's necessary. If you see topographical differences in your site, you might stratify and sample more intensively here and less intensively there. But yeah, it's a real balancing act. Marty, did you have anything to cover that?
Yeah, the only thing I'd add to that is this is just a proposed list of metrics or potential things that we could be asking for in Sustainable Wine Growing Australia. It's certainly not something that we'd introduce anytime soon, and there's no requirements for anyone to go out and do a whole lot of soil sampling that they haven't been doing before. So please don't be worried about that. It's just a recommendation for where we might go in the future, and it's definitely not something that's compulsory. But yeah, I think it's, if you're doing it already, this would just be I guess a potential repository for that information, and one way that you could track your data over time. Robin, do you mind if I jump in and answer another question, just because I need to leave? There was a question there, someone asking for the data on the emissions, the pie charts that I showed. What we can do is send you out the link to the full paper that's got a lot more of the information in it for reference.
Excellent, thank you. And thank you for your presentation today, Marty. We will say goodbye to you now, but we still have a fair heap more questions to go. So Michael Cutting, hello Michael, has there been any analysis completed on cover cropping practices versus irrigation intensity? So assume that there might be some trade-off between higher production, between the amount of irrigation water that you need to apply in these higher production areas like the Riverland. So you touched on it a little bit, Tom, but sorry, Joe, but if you could expand on it, that would be great. Thank you.
Well, for you, thank you.
Yeah, sure. Yeah, good question, Michael. No, we don't know. We don't have that sort of trial yet. It would be really nice to. I think some of the differences that we see with the medic and rye, especially, where we saw reduced anthocyanins there in the second year in Monash. We saw similar effects over in the Barossa. It'd be nice to be able to reduce the watering rates on those vines to see if we could perhaps bring those anthocyanins back up. If that's a factor, you know, if the berry size is actually contributing to that as well, I'm not sure. So yeah, very good question. One that I've been wondering for a while, but yeah, we don't have an answer to that yet.
Thank you. And we are going to have Jason Smith from CSU delivering a webinar in a couple of months' time, and he did work on the water use of different cover crops. So but that was in orange, so not in the Riverland. So there will be more information on that, but yeah, very keen to see you continue this work to find some answers to those questions. What was the input cost to apply the rye and medic per hectare, Tom?
The input cost, good question. I think, look, we've estimated it before, you know, a 30 kilo per hectare rate. You know, a bag of medic and rye is not very expensive. So you're looking at something in the order of, you know, a few $100 per hectare, and there's a one-off cost. So and then you can, you know, depending on where you're putting that, you're probably only applying that to the 30 centimeters directly underneath the vine. There's no need to go any wider than that. You could just get away with one strip as well, so one seeding line, and your cost then, you know, is decimated. So the cost of putting medic and rye in is really minor, you know, and the opportunity is pretty great there. So something I recommend for that sort of cost is, you know, you could get out there with a rake and put some in, you know, down one row or a few rows and see how it takes off. And, you know, the other thing I always advises to try and go for diversity. So you don't just have to use one medic species and one annual rye species, although I would strongly encourage you to put annual rye in and try and avoid, the more perennial varieties. And then, you can have a mixture of different legumes, and, whatever takes off is most suited to your soil. So, yeah, I think that kind of answers the question. Tim, sorry.
Can I make a comment, Robin, that
Yes,
touches on what Tom was just discussing in the earlier question about irrigation? What I've taken from, doing this work over the last 5 or so years is that not all cover crops are created equal.
Mm-hmm.
And so it's really, I think, as Tom said, it's important to get that diversity in there. It's also important to select cover crops that have desirable traits for your particular system. So that sort of try before you buy approach of, just try a little patch and see how it goes and see how it works with the system is really good advice. And when I think about the kind of traits that you might want in a cover crop, I imagine you want something that's grows actively in the winter when the water falls out of the sky for free, right? So it's not competing for irrigation water. It's something that sets its own seed. So as Tom said before, that it's self, regenerating, and then it dies off in summer to create a nice mulch layer, which then reduces evaporation, but it's also not actively growing when you switch on the irrigation water. So I think, just thinking through what are the desirable traits that you might like to have in a cover crop and then matching that to your system, I think that can be tweaked to deal with some of those irrigation related questions. But absolutely something we have been quite keen to do for some time is to do some trials on a larger scale where you can manage the irrigation of the particular trial rather than having smaller plots where we're dealing with, one block and one irrigation regime.
Yeah, okay, great, thank you. Just on the application rate per hectare, just to clarify, was that a rate per sown area or a rate per hectare, vineyard hectare?
Yeah, sorry, that's per sown area. So,
okay.
Yeah.
Yeah, okay, great, thank you. And just a question, and it may be for, either of you, but maybe Tim, going on from your last answer about the desirable traits, there's a question here about using clovers instead of medic. So is this something that you have looked at in your previous work, and do you think that you could get similar results by using clover instead of medic?
Yeah, that's a good question, and I'm sure Tom will chime in on this one as well with the results from the earlier work in other regions. Again, I think it comes down to, in some ways, talking to your local seed supplier and seeing what they recommend for your particular region. So, climate and soils and things like that, and then selecting something that's appropriate for your region and for your system. So obviously we've been working with the medic and rye, and that's been very, very successful. One of the great things about that particular mixture is you've got a nitrogen fixer and something that's not a nitrogen fixer. And we tend to find that, as the nutrients build up with the medic, they start to fall out of the system, and the rye comes in and starts to pick up some of that nitrogen. And then as the nitrogen drops back down again, the rye starts to fall down in its abundance and the medic starts to take off again. So you get this sort of cycling that goes through this successional process. To my mind, I look at them both as nitrogen fixers. And so that would be the trait I'd be focused on. But again, talking to your local seed supplier about what would work well in your system and getting that local advice, I think is really important. But I think, Tom, you could probably add to that from the earlier work with the clovers.
Yeah, sure. A little point. I mean, we tried clover in the Barossa and Langhorne Creek. Chris Penfold was, had a mix of fescue and clover there, strawberry clover, and that the strawberry clover worked really well in the first year, and then after that the fescue took over and completely dominated from then on. So it really became a fescue trial as opposed to a clover and fescue treatment. But yeah, in some other work that I'm doing at the moment in almonds, we're trying a bunch of different legumes. In fact, actually, I wanted to refer to the Joe slide earlier, which had all the the highest carbon to the lowest carbon stocks across the Limestone Coast and Barossa. And in fact, the site with the highest carbon had had quite a lot of vetch in growing in the undervine space there. So, you know, vetch has different characteristics again, but the point is it doesn't really matter too much as long as, you know, you've got this balance and you're setting up an ecosystem, you're setting up a steady state, and hopefully that ecosystem is stable enough to maintain itself, and, you know, resilient enough to deal with, you know, any of the droughts and heats and floods and, you know, things that occur.
Okay, great. Thank you. Now I think time for just one last question. So Jason asked, he came in a bit late and asked about the sowing method for the medic rye. Can you talk to not just the method that you used, but other methods for establishing these cover crops?
Yeah, sure. So yeah, what I did for this plot, this trial, is I used this a market gardening seeder to put it in. But there are various ways. You really need, if you're trying to put it in the undervine space, you really need one line of seed along a dripper line. And if you can get that to establish in the first year, the second year, the medic and the rye will spread, you know, quite naturally and quite easily. So you can use a traditional seeder and just try and get it as close to the vineyard or to the vine row as possible. And if you can get it, you know, close-ish to the vine row, then it will spread the next year. We've talked a lot about using planting it in the mid-row and then getting 1 year of biomass and letting it set seed, and then using a side thrower mower to just side throw the biomass to the undervine space, and that should do the trick as well. You know, the beauty of medic and rye is that they're really hardy, really easily established varieties, whereas, yeah, trickier things that might not work for, but it's a good way to get started. Native grasses and native seeds are trickier again, but, you know, they're such fantastic things to have. I'd really love to do some more work, and, you know, we're in discussions with various people to try and figure out a new way to trial natives in the undervine space, or in fact the whole orchard, vineyard floor. So there's lots more work to be done in this space, but yeah, so I've gone on a tangent, but, you know, it's really interesting stuff.
It is great, and thank you all for the work that you've been doing in this space. We had around 400 people registered for this webinar, so it just shows that it's not just you guys that are really passionate and interested in this, but there's a lot of people in the industry as well. So thank you all for your work and for sharing your knowledge and experience so generously. And thank you also to everyone, the audience, for participating. Your questions are always fantastic. It's great to get that interaction from you. So as a reminder, the webinar has been recorded and it will be emailed out to you. We'll make sure you get the links to the YouTube videos that the guys have produced, and the article that Mardi mentioned as well. So the next webinar is on Thursday, the 17th of August, and Dr. Mardi Longbottom will be joining us again from the AWRI. She will be talking to us about greenhouse gas emissions and more, so digging about in the Sustainable Wine Growing Australia data. So if you'd like to register, you just need to jump on to the AWRI website, and we look forward to seeing you again next week. Thank you to Tim, Tom and Joe, and to Mardi, who had to leave early. Thank you.
Thanks, Robin. See ya.
Thank you.
Thank you, Robin.